So when you see it go red, we're live, and whatever's in the red box is. Hi, welcome back, everybody. So this is Martin Leahy, and welcome back to the next installment of the Biophotonics and Imaging Graduate Summer School. And this installment is on brain biophotonics, and we're going to have Professor Arjun Yodh from Penn University. And he is going to tell us about neurophotonics. So over to you, Arjun.
Okay. Oh, so now I should see it live on me. Yes, I see. Okay. Hi. So, thanks everybody. Thanks, Martin, for organizing everything. And I wish I was in Ireland. You owe me some Guinness or something like that. So I guess I did, I did change a little bit the nature of my lectures. Of course, it was heavily focused towards brain and neurophotonics, but it was also heavily focused towards diffuse optics. And in fact, I would even almost say the first 3 lectures had to do with the nature of the measurements, how to understand them, what are the limitations, how can you use them, and so on. And then the 4th one had the many more sort of application example after application example, and I tried to at the very end say something about the microscopy, what's happening, but the emphasis really was on diffuse optics. And so if you're hoping for a lot of microscopy, I apologize for that. I'm happy to talk about it. And let me also say, just feel free to ask questions about any of the 4 lectures. Don't feel like you have to ask only about lecture 1 now. Just whichever question comes to your mind, just feel free to ask about that.
Okay, so why don't we get stuck in with the first question? Meng Li in Lund, Sweden, is asking, okay, this is an administrative question. Will this stuff be available temporarily? And yes, it will be. Ate Kazari at Twente in the Netherlands is asking, when measuring on the patient's head, considering the scalp, skull, and the brain, how can we determine the relation between the source, detector distance, and the penetration depth? Yeah,
so that's an excellent question, and so I want to answer it with a few different answers. Okay, so the first point is that, loosely speaking, if we have an experiment, I don't know, can you see my hands? Maybe you can.
Yes.
Okay, so I have a source here and a detector here. So they're separated by some distance rho, say. For a typical tissue media, you can expect that your penetration depth, that mean penetration depth, might be ranging from 1/3 of that rho over 3 to rho over 2. But that, so if you have it 3 centimeters apart, then it can go in 1.5 centimeters on average. But then there's the width of that distribution is also about the same. Okay, so it's actually a pretty large range that you can penetrate, and you could penetrate, I would say, as much as 2 centimeters in. Okay, and probe that tissue. Now, the trick, though, as you've asked, is that when you do these kinds of experiments, the tissue is layered. It's not just semi-infinite. And so ideally what one wants to do is to detect signals from the cortex and not from the scalp and the skull. And so I spent some time in some of my lectures talking about ways that people try to ameliorate that problem. And so if you use the differential path length approach, then there's some models that you can have for what is the path, average path that the light goes, that you could put into a kind of modified Beer-Lambert law. If you use just the diffusion equation, then you can imagine having just, it's useful to have a short source detector separation and a long source detector separation at a minimum. And then you can use analytical models to distinguish the overlayer from the deeper layer. And then you can use things like modulation techniques, pressure modulation techniques, because you know that when you apply a pressure to the scalp and the skull, that the brain tissue flow isn't affected, but the overlayer is. You can use that to with some of the new analysis tools to hone in, even though you're integrating over that whole region. The fact that you have some that's penetrating into the cortex, and the fact that you do this modulation lets you select out that. Okay, so the short answer is, roughly 1.5 to 2 centimeters for say a 3 centimeter separation is the depth that you can probe, but you're integrating over a large region. And if you want to separate extracerebral from cerebral, then you have to do a little more work. So I'm not sure that answered the question, but hopefully it did a little bit. I can't hear you, Martin.
Sorry, I muted myself.
Okay.
Okay, so here we go. So let's have a look here. So in lecture 4, blood flow informed PDT. Can blood flow alone determine the amount of single oxygen? Shouldn't the fluence rate be controlled according to the photosynthesizer activation? Do you want to answer that question first? There is another one.
Oh yeah, okay. So let me just, before I answer that, just remind you what we were doing in that experiment. So first of all, it was an animal experiment. It was an experiment on a mouse where we had a tumor, a rift tumor, and we were doing PDT on the rift tumor, and then we could look at, did it work or did it not work? And then at the same time, we were measuring the blood flow. Actually, we can also measure the oxygenation, but the focus in that was on the blood flow, because it turned out that when you looked at the blood flow during the PDT, in the first experiments that we did, we were just surprised at how much it was changing. And sometimes it would go up, and many times it would be coming crashing down to, and essentially the vasculature is turned off then, if you like, and there's the supply of oxygen is turned off, and so the PDT would be turned off. And so at that point we realized, well, if all this is sort of happening in these experiments, then first, we could look at this vascular shutdown and see, is it correlated with the efficacy of PDT? And we found that it was. Okay, if you have a significant vascular shutdown during your PDT, the PDT is not going to do as good a job at killing the tumor. But then the second part, which is I think what is being asked, what we tried to do then is to use this as a feedback. So we'd continuously measure the flow, and if we found, and so we would try to do the measurement with high fluence rate. And that's the normal way that people do it, okay? But then if we found that the flow went suddenly low, then we would have a feedback to the laser, or a shutter in front of the laser, and basically lower the power of the laser to let the flow recover in the tissue. And so our hypothesis was that by doing this, you can sort of you can more effectively, keep getting enough oxygen that you need for the PDT process to work. And of course, this is going to vary with, and by the way, I should say, when we did the feedback, we didn't go to 0 fluence rate. We went to just a low fluence rate. And then when the flow recovers, and you can have to set a threshold for this, then you can go back to the high fluence rate. But basically, we looked at this in animal models. We're going to certainly want to try it in humans if we can. But basically, we've looked at it in animal models. We have looked at it with different PDT agents. And, we found that this approach actually generally is good, but of course it does depend on the agent. So I think that's my answer to that first part of that question. I can't hear you.
Sorry, I've been muting myself. So lecture 2, extrapolated zero boundary condition. 1 over ln, I think it says, this equation for fluence in semi-infinite media. Why there is no term addressing fluence shape variations in different turbid media? Are we approximating this by considering an anisotropic source with tissue-specific diffusion contrast?
Okay, so let me see if I understand that. Okay, so the question really is about an anisotropic. Well, okay, the question is about the extrapolated boundary condition approximation. And so, yes, okay, so let me first go back one step. So the mixed boundary condition, where the flux is proportional to the gradient of the flux at the normal derivative of the flux with respect to position at the surface, that's an exact condition, okay? What we did to get the extrapolated boundary condition is we made a couple of approximations. The first one is that we replace, so the source indeed is a fiber that shoots light in, and so the light will be, the light source, if you like, will be at the beginning when it's leaving the fiber and going into the tissue. It's going to be, you know, have a well-defined direction. Then it starts to scatter, and then it becomes randomized. And so what we're first assuming in this model is that we can replace the source with an isotropic source, one random walk step into the medium. Okay. Approximately one random walk step. So that's something that is often done, and part of the reason for that is that it's you know, we know all the solutions. It's very easy to model it for point sources, but that is an approximation. I will say that people have looked at the you know, danger of using that approximation, and it's not really too bad as long as you're not too close to the source with your detector. Okay. And so in these experiments, typically, you know, the random walk step might be a millimeter, and the source detector separation of the surface would be centimeters. Centimeters, you know, 1 centimeter to even, you know, 5 centimeters even. And so that, you know, that's many random walk steps away. And so in that regime, this approximation is just fine. You know, the other approximation that we make has to do with, you know, replacing the true value of phi with its Taylor expansion and just keeping the lowest order term. Okay, and that lets us use the boundary condition. So that also, that is a small approximation. I don't think that's a very significant one. And again, people have done Monte Carlo, people have done all, you know, people have even tried, you know, people have solved the transport equation for certain kinds of situations in one dimension. And these are, for the regimes that we're working with, that's okay. Okay, and again, but not perfect. And so you have, you know, you should definitely be aware that if you're making measurements close to the source, that you might want to think about getting rid of these assumptions. Okay, and then you're going to necessarily have to go to a more numerical approach, a more computational approach for the analysis.
So Monte Carlo.
Yeah. Monte Carlo, for example.
Yeah. Okay, continuation also from Ashwini in Sweden. Should we expect significant errors while plotting sensitivity function over penetration depth, as the analytical diffusion equation is assumed to have zero fluence outside the boundary? Many interesting approaches to imaging, how better to understand regularization effects. Not sure what that means.
Yeah,
you do. And they so.
Well, okay, let me maybe just take that piece. So there was two pieces there. So I would say, you know, the analytical approach with the extrapolated boundary condition, you know, you can have been compared in the simple regimes. Again, not when you're close to the source and things, not when you're too close to the surface. Especially the problem is when you're too close to the source. Okay. But when you're not too close to the source, it works pretty well. The sensitivity curves certainly don't change very much at all. I think the second question was about regularization.
Yes.
Martin, just remind me what it was.
The second part, as the how better to understand regularization effect. Is it Tikhonov the best approach as a starting point? And while reconstructing non-spherical inclusions voxel-wise, using 1D data, which regularization would be better to consider?
Okay, so there are actually, you know, again, I tried to emphasize this in my lecture. There's a big field in imaging that worries about this, and inverse problems that worries about what's the right kinds of regularizations to use and so on. And certainly Tikhonov is one of the most common ones, okay? I mean, basically it just depends on what do you think that you want to penalize what do you want to constrain? That's what it basically boils down to for regularization. Maybe you want to constrain large amplitude fluctuations, okay? Or maybe you want to constrain sharp changes in space, okay? That they can't be so sharp, right? So again, when we do like tissue phantom experiments, usually we do have sharp interfaces, but in tissue itself, it's probably not so sharp. And so so one has to think about that. So the Tikhonov isn't the final word by any means. People try different kinds of norms. And then we ourselves have found that some kinds of norms are better for scattering than they are for absorption, if you want to reconstruct. So that's a good question, and I'll just say that it's a subject of research. But if you just use the Tikhonov, you'll probably get reasonably good images, but you maybe not the absolute, fidelity in the optical properties, for example.
Okay, at Kazari in the Netherlands wants to know, when measuring on the patient's head, considering, didn't we already do that one? Maybe the next one, lecture 2, to what extent do the measured absorption and scattering coefficients change between different human subjects?
Yeah, that's a good question. So in the brain, so there's a lot of data out there. I would say that scattering could be changing, say the mean would be 10 inverse centimeters, so 1 millimeter, okay? But I could easily see it could be ranging between 5 and 20, 5 and 15 at least. So it could change a lot for different people, all right, for the brain. Even for the breast, it depends on how, fibrotic it is, for example. I think you have to be, it's better to be measuring scattering, frankly. A lot of times people are assuming it, but I think it's better to be measuring it. And similarly, the absorption, I would put the same sort of range of, even for normal subjects, the same kind of range. Maybe it's not plus or minus 50%, but plus or minus 30%, but there'll be outliers. So it's better to measure all these things. I mean, that's why when you're doing the measurements and you're only going for the relative changes, embedded in those measurements are assumptions about what the average optical properties are, which are usually not measured, or quite often not measured. They're usually assumed. And so it's, in my mind, it's better to do the absolute, at least, even if you're going to look for changes, it's better to do measurements to try to figure out the absolute, so you can put them into those theories.
Okay, yeah, for a sanity check, apart from anything else, I guess. Truly Tritarshi at Ervan wants to know, while a large portion of tissues can be thought of as either optically thin, like the vitreous of the eye, or optically thick, like portions of the skin, how do you distinguish between the two? Could you reiterate the different assumptions we have to make for the two cases?
Okay, Martin, I just have one question. Is it possible for me to see the question also? It should
sure,
be, right?
If you click on the chat box with the question mark, they should come up. And if you click on the published.
Right, I'm on that, but basically I just want to scroll down.
Yeah.
So how do I can't seem to be able to do that.
If you click within any of those text boxes and then press the down arrow, it should scroll down.
Oh, okay, I got it. Okay, so I see. That's helpful to me. Okay.
And yeah,
so I'm on
the published ones. Yeah.
So I'm on the one from Irvine, Truly Chatterjee.
Yeah.
Yes. Okay. Could you discuss? Oh, okay. So again, I guess at some level the question is, what kind of samples could we be looking at? Okay, and so I'd say the first thing is we want to be looking at. I mean, this is going to be useful for samples which are thicker than, say, I mean, you'll be happy if it's like 10 random walk steps. Okay, all right. So maybe a centimeter. All right. It's not like it's. I would just say that you could probably get answers, and we've done experiments in rats, right, and stuff like that. Even if you're within 3 millimeters of the surface or 2 millimeters of the surface, you're going to get answers, and they're, again, if you want absolute answers, they're going to be, you'll have question marks about how absolute they are. But if you're looking for relative changes, then all of the many of the systematic errors that arise from the diffusion approximation get divided out, it turns out, in practice. So I would say if you want to apply DOS and DCS and these things, you can apply it. It's better applied. The target tissue should be like a centimeter or so, or centimeter or bigger, for example. It can go to half centimeters, okay. If it's less than that, maybe you should be doing some other microscopy, right? Or, you know, so that's certainly, it's, and then there's this no man's land between 0 and, you know, 3 millimeters or 2 millimeters, you know, where you're using, there's all sorts of interesting techniques, photoacoustic microscopy, lots of things. And so, you know, the resolution will not be, will be degrading as you go deeper, but you can still get good, useful information. So I think when it's in the middle range, say between, on the order of 3 millimeters, then that's sort of the borderline where you should start really thinking about using other techniques. But let me just say, for materials beyond biomedical samples, yeah, the answer is yes. In physics, right, or material science, there are all sorts of soft materials that one is interested in studying, like colloidal suspensions, foams, sand piles. We're interested in what's the structure of these things, what's the dynamics of these things, and they're very fundamental questions about many body hydrodynamics. What's the effect of interactions in causing different structures to form? And we even, for example, many years ago, did some work with paper additives. Okay, so if you add this paper additive that this company was making, they added to a paper to make it waterproof, more waterproof, like on a milk carton or something. And basically, it was a turbid medium that they're, you know, when they make this stuff at the factory, it's a turbid medium that could make a gel, and if it made a gel, it's bad. So we did some experiments where we could determine, did it make a gel or did it not? So actually, you know, this conference is about biomedical application, but basically these diffuse optical measurements are good for looking at any optically thick medium. So any medium which is a 10 random walk steps thick or so is going to be game.
Okay, Giovanna from.
But let me just make, so microfluidics though, that's not so much. There's not so much scattering in microfluidics. Basically, those are pretty optically thin. I see the end of that question. Okay. Okay, go ahead.
Okay, Giovanna from Brazil wants to know from lecture 1 on absorption. You mentioned the extinction coefficient depends on the wavelength of light. Which wavelength has the smallest extinction coefficient? And wouldn't it be affected by the tissue structure? For example, tissue with a high presence of collagen versus high water content.
Yeah, so, well, okay, so what's the best wavelength to use? I mean, again, that's going to depend on the kind of tissue you're looking at, right? It's going to be different for breast. For the say what's the best wavelength to use to have the least absorption? Okay, let's say that's your question. So then, first of all, that depends on the tissue. So it's different. It's going to be different for the breast on average compared to the liver, compared to the brain, right? So generally, if you have more blood, then you want to be more into the near infrared, right? Because that's where the minimum is for the blood. Okay, and so, but I would still say that nominally around 800 is a good range to work, where you're going to have a fair, you know, depending on how much blood it is, you're going to have a fairly good. That's going to be the optimal range to figure out stuff about blood, but also to penetrate into tissue. For the scattering, I don't know, again, so I didn't emphasize this very much, but, you know, people have studied the wavelength dependence of the scattering, and it obeys a kind of a power law, right? So it's not like there's one sharp optimal wavelength to use in, say, within the near infrared region, or even going into the visible. I will say that it does go down. It can go down a lot. There's a lot of interest now that's been started to go farther into the infrared. All right, so don't just work, between say 800, 690 and 900. Say go farther, go past a micron, and then you have to just be careful about the water has a lot of structure. There's a lot more vibrational modes that could be having impact on your spectra, but you can find wavelengths where there's a relatively low absorption, and actually there's also low scattering, and so there's new opportunities in that regime for both diffuse optics, but also for microscopies.
Yeah, so I think 730 seems to be quite good if you don't have too much blood, right? That usually shows the best penetration depth. But then if you go to 1064, you're not so sensitive to blood. You're not so sensitive to melanin, which is quite important. And the other thing is you can use probably 4 times as much light, because now your limit is the infrared damage as opposed to the visible light damage. I guess what you lose is the low cost of using a silicon system. Actually, one other curious thing that people forget about is you get an extra 6 dB by going to in-gas detectors, right? So silicon detectors at those kind of wavelengths have quite a poor quantum efficiency, whereas you get much better. You can get up to 90% quantum efficiency with an in-gas detector. So all those things being equal, it really depends. If you have a very bloody tissue, then probably almost definitely the infrared is your way to go, or 1064. You also have a nice laser there, or very well developed lasers there with plenty of power and reasonable cost and reliability. So that's what I would advise people. If you have a more or less bloodless tissue and there's not too much melanin, then probably 730, especially if you want to build a cheaper system with silicon, is good. Yeah.
But I would also say, even beyond 1064, there's actually been a lot of development of contrast agents, even in the maybe around 1500 or 1100 or so, or between 1100 and 1200 now. So there might be opportunities to look at those if you're looking at contrast agents, because they're less scattering. So I mean, again, you have to look at what your application is, but there's been a huge amount of work in the near infrared, and so there's a research opportunity, I would say, to go farther to the red than has been done in the past.
Okay,
for all these reasons Martin said as well.
Truly in Irvine wants to know, could you discuss the potential applications of diffuse optical spectroscopy for general scattering materials beyond biomedical samples? I think you've mentioned that already, and or DCS for applications, for example, in microfluidics. So I think you touched on that.
I think I answered that question, but I'll just reiterate. The answer is yes. There's opportunity in all sorts of soft matter systems like foams, colloids, gels, sand piles, lots of different kinds of systems where you can't look inside them, right? And you want to figure out what's happening structurally and dynamically. And so both diffuse correlation spectroscopies and also the optical spectroscopies are useful in that context. For microfluidics, I think unless you have a network of microfluidic channels that have lots of scatterers in them, that's the example where I could think that it could be useful. If it's just sort of a few channels, then I think you're better off just doing microscopy.
Okay, I think when we swapped over to you reading them, we might have missed.
I think we missed a lot. Yeah, sorry about that.
Also by a Truly. So let me run it by you. You can tell me if you've answered it. While a large portion of tissues can be thought of as either optically thin, didn't we discuss
no,
that?
we discussed this one, yeah.
Yeah. Okay, very good. So let me go back down to Christian Fernandez at Tufts wants to know, can you discuss when you would want to use higher order PN approximation over P1, and what situation is P1 perfectly okay?
Sure. So the situation where you want to use higher order is when you're making measurements closer to the source. Okay, that's the short answer. So if you're 10 random walk steps away, pretty much P1 is pretty good, right? That's the biggest situation where you'd want to go higher. But there are other ones. So if you have a very highly absorbing material, so the absorption is comparable to the scattering, like in the liver or something, then you aren't going to just want to, you're going to have to look at the transport equation a little bit more carefully. You can't just make the blind some of the approximations that you make when you go to diffusion. So you'll need to sort of look at the problem again but it's tractable, I think, for when the absorption is comparable in magnitude to the scattering. Those are the main application areas that I think that you'd want to think about going back to the transport equation and looking at it again.
Okay.
Let me just say one other thing. So all of these theories that I presented really are assuming unpolarized light. Okay, and so of course that's one of the interesting things about light, and you can learn a lot from the polarization. So you can actually formulate the transport equation even for the Stokes vector kind of. You can do a Stokes vector analogous formulation of the transport equation, which is obviously much more is even more complicated. But again, the where you'd want to worry about that is going to be when you're close to the sources.
All right. Wajahat in Oslo wants to know, while using continuous wave NRS, how can we calculate or integrate values of mu A and mu S in MBLL? There's a lot of acronyms here, because we cannot directly measure mu A and mu S while using continuous wave NRS.
Yeah, I didn't understand what the middle acronym, MBLL, what was that?
Yeah, I didn't get that either.
But let me just say, I'll make a comment. So I would just say that if you're just using CW, you are kind of stuck. Okay, I mean, you can use a lot of wavelengths. You can use broadband to constrain a lot about the absorption, and that could be helpful. So if you use broadband CW, then that's better than just using a couple of wavelengths. Okay, but really, I would recommend, if you're going to do CW measurements, make a measurement that's not CW first and get an estimate of what the average optical properties are. That could be time domain, that could be frequency domain. Then you can put them into your models that you use to figure out changes with CW data. That's what I would recommend. So, and that's what we would do in research.
Okay, Ming Li at Lund wants to know, tissue optical properties can be measured by several techniques. Which is the most effective technique in your opinion? For example, between time resolved technique and spatially resolved diffuse reflectance technique, or integrating sphere. I guess they're all kind of difficult, in my view.
Yeah. I mean, so again, that's true. And at some level, it's what I personally always like, the frequency domain. Okay, and that's just probably only because I started doing experiments in the frequency domain. And so I just got knowledge of all the little things that can optimize it. Okay, so but on a sort of a per source detector pair information, if I just sort of classified it, there's more information in the time domain than there is in the frequency domain for a single source detector pair, right? It's like the frequency, the time domain has it's the Fourier transform. It has all the frequency components, so not just one. However, of course, you can also scan the modulation frequency if you're doing a frequency domain measurement. Bruce Stromberg sort of pioneered that at UCI. But I think, so I mean, I would just say between the two of them, it's a trade-off, because if you're doing usually if you're doing a frequency domain measurement, you're measuring all of it. You have a lot of light, and you're not doing single photon counting, and so you're sort of you're integrating over a bigger signal, and then you just have to pull out the small change. But if you can, if you have to make a measurement with just one source detector pair, I think it's probably better to do it with a time domain. If you can afford to make several positions, then it's probably cheaper and easier to do it with a frequency domain. But then you also run the risk that the tissue property could be not perfectly homogeneous.
Okay, I think MBLL could mean modified Beer-Lambert law. Is that possible?
That could be right. Yeah. So, well, I think my answer still stays the same. The modified Beer-Lambert law, you can apply it to the time domain as well. Usually, people are now using the moments. There's some information in the moments, which so you get a photon distribution in these time domain measurements. So there's a mean time and there's a variance and so on. And folks have done some nice research on this problem, and it turns out the variance can probe a little bit deeper, for example, than the mean time. So there's actually some other little tools, but still all of them can operate within the modified Beer-Lambert law. All of these approaches can, because basically that's just a Taylor expansion. And okay. Let me just make one cautionary note about the modified Beer-Lambert law, which happened to me. That's the reason I'm making it. It's just that remember, it's a Taylor expansion, so there have to be small changes in absorption. And so we were doing some experiments with Keith St. Lawrence where we injected a bolus of ICG into the brain. And so we measured the absorption change there. And by measuring the time dependence of that, you can back out another measurement of the flow. But in fact, if the change in the absorption is large, then you can't just use the lowest order Beer-Lambert. And so we found that was a problem. Okay, so if your changes are small, you're fine with it. If your changes are large, you have to be careful. You might have to use the next terms.
Okay, Hampus at Lund wants to know, in the first lecture you mentioned that we can measure the differential scattering cross-section. What can this knowledge of this parameter give you looking at tissues?
Yeah, so I wanted to introduce the differential cross-section but I introduced it really in the context of single scattering, and then I used that in the transport equation. And so it's important because basically that's telling you it's related to the anisotropy of the tissue. The G, there's a factor that was G. It was also on some of my slides. It told you how much the photons scatter in the forward direction when they scatter. And if they tend to scatter a lot in the forward direction, then the random walk step tends to require many scattering events to randomize its direction. So what you're basically one way to rephrase your question is, if you could measure what does tissue anisotropy tell us about tissue, right? And so I would say that tells us something about the nature of the scatterers. What's the size distribution of the nature of the scatterers? So if they tend to be if the G factor tends to be, and this is related to that form factor, it's just the average of the cosine averaged over that form factor. If the G factor tends to be small, then we know the scattering is fairly isotropic, and you might think it's more from mitochondria than something like nuclei or cells. Okay, so there is information there about the G that you can learn, and I suspect that people have tried to look at near surface tissues and the G as a way to diagnose different kinds of skin issues. But I don't have it off the top of my head, honestly, what they found. But the kind of thing you would find would have to do with the size of the scatterers, for example, or the index for refraction difference between the scatterers and the surrounding tissue, or the shape of the scatterers.
Yeah, so thank you for that. Louis Halleman in Rochester wants to know about the so-called modified Beer-Lambert law. You mentioned in those slides that the scattering coefficient variation is negligible compared to the absorption coefficient variation. What is the physics behind this? I mean, to gain some intuition.
So actually, I meant there's two things. I pointed out that if you want to make the approximation that everybody's using, then you first of all have to assume that the scattering is small. Scattering changes are small. That's not necessarily true, right? That's an assumption of the theory. So first of all, it's better if you know the mu s prime, because even if you have a rough estimate of the mu s prime, that'll give you a better value for this differential path length. But there is a term that's related to the scattering changes. It's smaller generally than the absorption term because there's a factor of essentially mu a over mu s prime, right? So it's probably smaller by a factor of 10 at least from the prefactor, but that doesn't mean that there couldn't be an effect. But so the reason that it's not a bad approximation is because if you look at that slide, which I can remember in my head, but I can't put it up right now, there was a term that had a differential path length L times some delta mu a, and then there was a term that had a differential path length L times the factor delta mu s prime times a ratio of mu a to mu s prime, okay? And so the fact that you had that ratio there makes that term smaller if everything else is about the same. So it's not a terrible approximation.
Okay. All right. In the experiment in lecture 1, you get 2 lines of phase and amplitude over distance, and can from the slopes find scattering and absorption parameters. How do you measure the data points that give these lines?
Oh, so in that experiment, you know where your source position is and you know where your detector position is, right? So there's a source detector separation. And so we make measurements at different source detector separations and then plot how is the amplitude changing versus source detector separation and how is the phase changing versus source detector separation. I mean, actually the phase was one of them and the other one was like the log of the distance between them times the function, okay? But anyway, some simple function of that measurement. You have to make measurements at many different source detector separations in order to get those curves. And that's related to somebody's question earlier. Yeah.
Yeah. Wajahat in Oslo wants to know when the target is bone tissue interface in continuous wave NIRS. Do we need to consider the mu a and mu s values of tissues only? And what about skin and fat absorption values, or do we neglect them?
Well, in practice, people basically are neglecting them, okay? So you have to, if there's, in principle, right, if there's a little bit of difference in the optical properties, if that's the only change, then you can handle the problem as a layer, piecewise homogeneous, and then you just have a continuous fluence rate and a continuous normal derivative across the boundary, and you can solve that problem. But another thing I'll just mention that could be, so it's not, the bottom line is that I don't think that the boundary conditions for that problem from one turbid media to another are really that critical, and they're nowhere near as important as the air tissue interface or a water tissue interface. But they, you can put them in, and people put them in the analytical solutions, for example, and you can put them in your numerical solutions. Another thing that is not usually mentioned very much is the index of refraction could be different, and that could be put in as well. Okay, right, and that'll have an effect. But again, I would say, and any people that are doing brain studies are usually not putting those things in. They may be fitting to a model that is effectively including a boundary condition associated with that, but it's certainly very, very uncommon, I think, for it to put in the index of refraction.
Right, but for other imaging techniques, that's probably very more important, let's say. For these diffuse techniques, is it more to do with getting quantitation that you want to put those in, or it's not about imaging, is it? Or
so if you're doing
it's about
if you're doing like
measurements.
if you're doing layered, so let's just say layered tissue, that's like a bone, then I would say it is better to put it in. And so if you're using the analytical or numerical solutions, you would put it in. That would be part of the solution. Okay.
Okay.
So basically the continuous fluence rate and the continuous normal derivative with the, times d on both sides.
All right, Luis Aleman in Rochester again wants to know, when you show and mention some of the breast cancer images using diffuse optical spectroscopy, what is the resolution achievable, and how is this compared to other techniques, particularly ultrasound?
Yeah, so I mean the resolution typically, I would say the best resolution you're going to get is a 0.5 centimeter. Okay, but more likely most of the measurements are maybe of the order between a 0.5 centimeter and a centimeter. So if your objective is to resolve a small tumor, this is not the choice for you. If your objective is to look at the functional properties of the tumor, right? Say, for example, in response to some chemotherapy or something like that, or to sort of more functional diagnostic property for characterizing it, then I think that still can be quite useful even for the bigger tumors. So the ultrasound can detect smaller things, but it generally, so ultrasound, right, is requiring a big contrast in the acoustic impedance, if you like. And so that's good for detecting the cyst compared to a tissue, right? It's not as good for tissue against tissue. But it's, if you want to go for, in terms of resolution, I would say you're going to definitely do better with ultrasound, if you can see it. There's some nice work actually by Qing Zhu, she's at WashU now, where she actually combines the ultrasound with the optical in breast cancer measurements. And so that's really nice because she can use the ultrasound to get better morphological information about the tumor, its location, and also its dimensions. And then she can take her optical data with that constraint and assign a better functional property, if you like, associated with the hemodynamics.
Okay, was that linked in with Bruce Tromberg's work, this multi-site study on, I think, neoadjuvant therapy? And what's the latest status of that? Is that?
Yeah, so I guess I'm part of that that network, and so that was not the last experiment I just told you about by Qing Zhu with the ultrasound and the optical. That wasn't part of that network. However, the status of the neoadjuvant chemotherapy, in my view, is it's encouraging. The results were encouraging, especially if you analyze it. So the most superficial way to analyze it was marginally encouraging, I would say. And the goal of it, just again to remind people, this was a study where women that have a breast cancer are undergoing neoadjuvant chemotherapy, and what we want to do is to make measurements sort of before, in the first week, in the middle of the process, so that's after a month or so or 2 months, and then towards the end, right, but right before surgery. And the idea is to tell whether the chemotherapy is working, okay? So you look at these optical properties, and you also look at the probability of chemotherapy response, right? And so what that study did is it looked at the midpoint and looked at the optical properties and compared people that responded to people that didn't respond. And it found it was at the margin with the p-value. It was a little bit less than 0.05, but in the number of, it was about 30 or 40 patients across the US, though, with a lot of different centers. And it found that there was definitely some, there was a value to knowing in the middle point that you could, that this chemotherapy wasn't working or this chemotherapy was working, because then you could change it in principle. But since then, we've really analyzed the data with Bruce, and with a different approach that essentially is using a Z number. Okay, so it's sort of normalizing by the patient's own normal properties. And in that case, we could find within 1 week the oxygen signals, or the oxygen saturation signals, the HbO2 signals, are really indicative of whether the tissue is responding to the chemotherapy or not. But anyway, going back to the question, the big question really is, no, the resolution is not as good as ultrasound generally. It's not as, if you want to do MRI, that's got better resolution also. But there's certainly some times, like X-ray, that X-ray misses tumors, and there could be tumors that you could catch with optical that X-ray misses, especially in fibrotic breasts. So there's niche opportunities in terms of screening, but I think the bigger opportunities have to do with functional diagnosis. And then, you're going to be still looking at things that are 0.5 centimeter or larger, usually. For detection opportunities, you have to use contrast agents. There is opportunity there, but because you can get a much higher contrast ratio for detection.
Okay, on the neoadjuvant therapy, while you brought that up, or I brought it up maybe, you talked about oxygen. So, but presumably you don't mean static measurements of oxygen. You're looking at changes.
Oh,
is
yeah.
That fair?
So, okay, yeah. So all of these measurements that I'm talking, almost all of them, there's a baseline period, and then there's a measurement outside of baseline period. So for the neoadjuvant chemotherapy, the baseline period is like before the treatment starts, right? So you're looking at a change with respect to before the treatment starts. But within 10 days, if you look at the Z number, if you like, the Z score, we define this because you made measurements on normal breast and the tumor region of the other breast. If you look at those Z numbers, then you can actually have a very good sensitivity to whether the tumor is going to respond or not to the chemotherapy.
Okay, let's take one more question, and maybe then we can give you a break for a little while. Are we due a break now? Somebody will tell me if we're not. So thank you for your interesting lectures, as many people have already said. During the presentation, Julia from Milan wants to know, you talk about absolute optical properties values. However, different research groups usually obtain different results. Using different systems and different data analysis tools, how important is the detailing the standardization in this? And I know, I guess there's also a need to realize that all of these things, are different in the site and the subject that you measure. So we will never have absolute measurements, right, that are going to be correct for the instrument, and the instrument is just calibrated. I'm guessing, right? Yeah, but maybe you want to answer that.
Well,
question.
Okay. So there's a couple ways to. So there's a European group originating from this bitmap grant, which I think is coming to an end, or maybe has already come to an end. But basically, part of the goal of that was to do this comparison that you're talking about. So it's an excellent question. And so, the easy thing to do is to make a phantom and to pass the phantom around to different groups. And so then the same phantom at least is being measured and by different techniques at different places by different people. And I would say that the phantom measurements are not too bad. I mean, they're not perfect, but they're pretty narrow distribution of variation. Okay. So then, but in practice, yeah. So I would, so that's not the same as doing it in humans. Okay. And so I guess ideally you could have the same human subjects measured by all the different techniques. Probably that would have to be in the same place. That's a good thing that would be interesting to do and find out. Personally, I think that would be a good thing to do. That's an excellent suggestion. I don't think that is happening. So the bottom line then is that we're relying on the fact that pretty much the phantom measurements are giving pretty good getting pretty good agreement among the different techniques. Not perfect. And so then we think that we just calibrate with that phantom and make measurements on humans, and then we can look at then there's probably a bigger heterogeneity in the human population measurements, so there'll be a bigger distribution from that. But without question, that's a good thing to do, and to check, for sure.
All right. Great. So we're live with Professor Arjun Yodh from University of Pennsylvania. My name is Martin Leahy from NUI Galway. We're talking about neurophotonics, and we will be back in 30 minutes. So 15:30 UK Irish time. Just Google Galway time or Dublin time. Thanks.
So, Martin, then 11 o'clock my time. We'll be starting again.
30 minutes from now. I've forgotten how many. Are you 5 hours behind us?
Right. Yeah. So it's 10:30. 10:30.
30 minutes from now.
Okay. So I'll just leave my computer like this, and then come back, and it'll be up and running, or I'll come back 5 minutes before that. Okay, and we can check. Yeah,
we're staying live, so you can just mute your microphone if you like, or even turn off your video if you like to. Okay, but we'll keep the channel live, so the questions keep coming in. Yeah.
Oh, I see. So, is it going okay? Is this going okay?
Yeah, sorry, I muted myself. So yes, it is. Yes, sure.
So this is the.
Yeah, it's going great.
Okay.
You're happy with the questions? We have lots more questions to get through. So
yeah. I'm glad about the questioning. I'm glad there are a lot of questions. It's interesting. Yeah. There's a lot more than I thought would be about the first lecture, because that was somewhat technical. But I guess people want to know the answers to those things. So,
but people are building systems,
But you can, but from your perspective, it's not like my answers are coming across okay.
They're coming across great. You're giving plenty of detail and quite sympathetic, which to the students, as we always should be. So that's fantastic. Yeah.
Okay.
My only concern is that maybe we won't get to all of them, so just keep that in mind. Maybe we can try to keep them shorter if we can, and I'll try and say less as well.
Okay, no, actually, I like it when you say stuff, so I think it's better. Actually, so don't say less. Okay. All right. So
Thanks,
I'll
Arjun.
take a little break then. Thanks.
Bye. Okay, welcome back, Arjun.
Great.
All right. Yeah, so maybe Seren will ask some questions during this period, but I can kick off with the first question. Let me just find out where we were. So, yeah, so I think the next question was from Meng Li in Lund. How do you achieve semi-infinite or infinite medium in real life? In our lab, we wrap the sample with a black sheet of tape, but I believe there will still be light bouncing back from the interface, and there is light leakage if the sample is not fully secured.
I mean, that's a challenge. That's one of the challenges of these measurements, and it's certainly, even that experiment, right, is not semi-infinite, it's layered. But so, you know, we do the same thing you do. We do our best to hold things. So for the head probes, right, we put the probe on. We're working on a lot of little details now to try to make like kind of a hairbrush mode of it, with, you know, some pegs that are to relieve the pressure. There are fibers that are coming in, but there are also non-fibers that are really, you know, sort of taking some of the pressure stress off when you push on it, and then you wrap it. So that's there's no way around that, and for the brain. And so again, it's not a precision measurement per se. I mean, I think again, if you can focus on looking for changes from baseline of something, that's always better. But if you can't, then still I would say that you're going to get a reasonably, you know, within a plus or minus 10% number if you try to do a good job of holding it. So I don't know if that's answering the question or not, but the answer is it's not easy. Right. Yeah, and so and frankly, even we did breast measurements where we have two parallel plates that come to the breast surface, and, you know, they're butted up against the sources and detectors that are in the plates. In that case, actually, we can have intralipid as a background medium, so the breast is in the intralipid as well. But you know, it's not perfectly slab in that case, but it's worked pretty well. I mean, it's the bottom line.
Okay, very good. And I guess as physicists and engineers, one of the first things we have to get used to is the idea that 10% or 20 or maybe even sometimes 50% accuracy measurement, order magnitude measurements can be useful in clinical situations.
Absolutely, yeah. So the thing is that, yeah, it's not like a precision atomic physics measurement when you're measuring a frequency of some atom or something, right? This is something where if we find out that if the hemoglobin is 30% more concentrated, or there's 30% less saturation, that means something.
Right. Yeah. So Anand is going to take over and ask some questions now. Over to you, Anand.
Okay, thanks, Martin. So the next question is, what are your thoughts on slop-based approaches to suppress signal from superficial regions of a diffuse medium? That is, subtracting a short distance measurement from a long.
Yeah, so that's the I'm in favor of that as a first step, and that's, you know, in a way, when you have a short detector and a long detector, that's kind of what you're thinking you're going to do. However, if you just model it a little bit better, you know, with 2 layers even, then I think you can do. You still want to have a short and a long separation, but I think you can do a little. You know, you don't want to just directly subtract it. You just want to do a little bit more sophisticated subtraction. So that's a good technique.
Great, thanks, Arjun. So the next question is from Hani from Finland. That's quite a long one. To measure oxyhemoglobin and hemoglobin changes, we need to use at least 2 wavelengths. I would like to know how the instrumentation and calculation process for the continuous wave, Fourier domain, and time domain. For both CW and FD, the light sources are continuously on. So how can we separate signals from different wavelengths?
Oh, good. Okay, so people typically do 2 things. One is that they have a kind of a shutter, so they do the measurements in sequentially, but you know, with a very short delay in time. So that's one way to do it. So for the frequency domain, you know, we would turn on one. You know, you're not usually going to turn on and off the laser. You'll just turn on and off a shutter in front of the laser. Or you can turn on and off the detection. Either way, one detector could be designed to detect one wavelength, another to detect another wavelength. So that's one way to do it. If you want to do it in parallel, though, you can by modulating the light at slightly different frequencies in the frequency and even for CW or frequency domain. So then you can demodulate it at the different frequencies. That's something people do for CW especially. And it doesn't have to be a high frequency modulation. It can be kilohertz or something. For time domain, again, people, one of the tricks that people do is that they have, if they're going to have pulses from different lasers at with different wavelengths, then they shift them in time slightly, so that for the same overall pulse, you'll get 3 different time of flight curves, or 2 different time of flight curves, depending on how many wavelengths you're using, and they'll be slightly separated so you can resolve them. Another trick that people are doing nowadays, which we're doing, for example, is you do a broadband. Okay, so you can put in a white light pulse and then collect all of the wavelengths using a grating spectrometer. And then you get a time resolved pattern for each wavelength that way. So there's a lot of different ways that people do that.
Great. The question continues. For time domain, I suppose the receivers use a kind of multiplexer to select the wavelength of interest. Is it correct? If it is correct, then there must be a delay time between 2 receiver activations. How should we calculate this delay time? If my assumption is wrong, then what is the correct configuration?
Yeah, I'm not actually sure that I totally understand the question. So, okay, as I said, there's one way that people do the time resolved. Okay, so you could time share 2 lasers. So one is on for a second, then you make a measurement. Another one's on for a second, and then just toggle between them. Another way that people do it, as I said, is that they can time delay one laser with respect to the other one. And then they just have the same, they do the same time of flight measurements, and that they can actually get the 2 pulses and resolve it. And the other way that people do it is they do this white light trick. So then you take the pulse that you get and you spread it out over a CCD. You have it hit a grating, diffraction grating. It'll spread out the wavelengths, and then you detect with a kind of a digital detector or some kind of a CCD the photons in each area. So those are the kind. That's a kind of more expensive approach, but that's a good way you can do it in parallel.
Thanks, Arjun. So the next question is from Priyanuj from Kolkata. I have a few general questions. What factors do you consider when adding relevant components to these models? Because they have to correlate with the optical experiments and theory. Also, another question, do you incorporate a systems biology approach to these physical models?
Okay, I didn't understand the first question. Did you understand it, Anand?
Not quite.
So you want to say it again?
Yeah. What factors do you consider when adding relevant components to these models?
But when adding
He's
what?
talking about the intensive use of mathematical models from your lecture, but he didn't specify which slide or anything.
Oh, okay. Well, okay. So that really depends on the experiment. If it's a monitoring experiment, for example, compared to a tomographic imaging experiment. So I mean, I guess for all of them, to me, the underlying differential equation that we're using and boundary conditions are from the diffusion equation. And you know, there's some level of constraint that we want to always put in. Okay. So we have some expectation of what the numbers are. That's one constraint. As I said earlier, you can have a constraint about how large a change you expect that you would ever see, or how fast that change occurs in space that you would ever see, and you can put those into your model as well. I'm not sure if that answers your question, but those are the kinds of things that we would put in. Of course, if you have many wavelengths, that's very attractive. So even if it's not broadband, but if you have like 6 wavelengths, then really often the number of unknowns is like the chromophore concentration and the scattering. And if the main chromophore is just oxydeoxyhemoglobin, then you overdetermine the problem if you use a lot of wavelengths, and that can help a lot.
Thanks for that. The next question is from Shanavas. How can we avoid optical application in dentistry, and how we can handle the error that occurred due to the movement while scanning tissues?
Okay, so in dentistry, I think that diffuse optical, maybe it could have some value because the teeth is fairly scattering. I don't know of any. I don't know, but I haven't checked everything. I don't know of any experiments along those lines. But there has been a lot of biophotonics experiments in the dentistry. I think OCT is one of them, techniques that are used, microscopy techniques, to just try to find disease, looking at near surface tissues and the spectroscopy of them and morphology. What was the second question?
How can I handle the error that occurred due to the movement while scanning tissues?
Okay, so if you're scanning tissue, you know, there's always some motion. And so the easiest way to solve that problem is to measure fast. Okay, well, it depends on, again, you have to measure significantly faster than the time scale of the motions. So at least then you could resolve the motions, and you can, at the very minimum, decide that some data is bad because of motion and throw that data out. So we have, so one approach that a lot of us in the community are doing is just try to make measurements faster, right? And so I think with the DOS measurements or NIRS measurements, you can make them pretty fast. I mean, you know, certainly way faster than the heartbeats. For even DCS measurements, the flow measurements, we've now gotten it up to like, you know, depending on the signal to noise, up to even like 50 hertz measurements. And so then in principle, you can, by measuring fast enough, you can sort of catch the motion artifacts and not use that data. You can also think about having an ancillary measurement, which is looking at motion. Okay, so again, if you're measuring an exercising patient or something like this, or you can put an accelerometer on and capture how the tissue is, you know, if it's moving and if there could be a motion of the probe with respect to the actual tissue you want to measure, which would cause a systematic. So those are so to deal with motion artifacts, the first thing I would say is try to do a measurement in a situation where it's going to be small. And then the second one, make measurements as fast as possible. And then the third one is make measurements of the probe, put in ancillary instruments so that you can tell the relative motion of the probe compared to the tissue. And then at least again, both of the last two methods offer a way to systematically exclude data where there's a motion artifact.
The next question is, for the modified Beer-Lambert's measurements, you get an absorption changes over time. Could you also get the baseline absorption? Is it sufficient to just add a detector at a different distance?
Yeah, so you can try to do that, but you'll have to assume something about scattering. But you could assume you'll have some, it won't be as reliable. A multi-distance CW measurement can give you some estimate, but it has some assumptions in it usually.
Okay, great. Next question is from Atta Cizari from Netherlands. We have performed laser spectral contrast imaging on the live chicken embryos, and we have performed separate measurements on microtubing flow phantom. Would it be fair to try report absolute blood flow values of the in vivo measurements based on our observation of the flow phantom experiments?
I think that's a little bit risky. I mean, that's a good confidence. I guess ultimately the answer to that question depends on how much you believe that the flow phantom is reflecting the in vivo case, right? And so I would be a little bit nervous about that. So if you want to do a calibration, then I would say the kinds of calibrations you can do are, well, one that you can do, which I talked about, is you can inject a bolus of a dye, some contrast agent that you could detect optically. And so then you can, by measuring sort of the uptake of that and then the time scale on which it goes away, you can make a simple model to estimate flow. And then at the same time or right after or right before, do the measurement with the speckle contrast. And then you'll have a calibration. So that's, an in vivo calibration. I don't know if it's possible in every situation, but it may be that you could do it in some, you don't necessarily, it's if you can, it's better to do it in the same, if you want to get absolute flow, if you can, then it's better to do it in the same living thing. And that's what we did in our experiment that I talked about. But even if you do this measurement of the bolus flow in a lot of different rats or chicken or whatever you want to use, or human, then you can average over those, and so you can get some kind of a rough calibration for absolute flow.
Okay, Luca from Strasbourg wants to know, what are the best detector candidates for time domain DOT or DOS? Why is it so hard to find commercially available silicon photomultipliers modules for single photon counting applications with high time resolution?
Yeah, I'm not the greatest person to answer that because I mostly have been doing frequency domain. So I would in fact encourage, I think that there's some good reviews about time domain. Either, the work that's coming in from the Milan group, and also there's some groups, Ilias, Taxidis in UCL, may have better discussions of it than I can give you. I mean, I guess ultimately the reason so for example, for the DCS detectors, which I know much more about, it's hard to get photon counting low noise detectors in the near infrared. And part of the reason that it's hard is because it's not lucrative. I think that people can make them for sure, but it's not financially lucrative for a company to make a lot at this stage. So I guess what we need to do as a community is to keep doing our research experiments to demonstrate the value of the information that we're getting. And then that can help to try to convince these companies that make detectors to you know, obviously if there's easy solutions, they would do it. But if they work a little bit harder, or they make more detectors, they spend more of their effort to make detectors in this range, that could be motivated by showing clinical value. So I don't know if that, again, that's probably not answering the question very well, but that's why it's hard.
Thanks, Arjun. Yanis from UK wants to know, what are the challenges in the translation of a diffuse fiber optic probe from ex vivo into in vivo?
Okay, the challenge is it's not just the probe, but certainly the probe, you know, ex vivo is sitting still. That's a much easier measurement to do. You can take it longer if you want, but you can also set up the device so that you're, you know, everything, all the detectors are in the right positions and so on. If you, once you go to the in vivo, first of all, there's usually a time factor. Well, unless you're really going to make continuous measurements at the bedside for a long time, then you just have to worry about if there's changes if the people move. But there's always sort of a time factor and a factor of motional artifacts that someone also brought up, which makes the measurement harder. That's the biggest thing. Holding the probe on a person is much harder than just putting it on a phantom. But the analysis also is harder because really the and the name of the game in the analysis also is to get as much other information as you can, because putting it on a head, putting this probe on the head is not a semi-infinite medium. It's not even a, you know, a sort of an infinite set of parallel layers, right? So the analysis itself is approximate, right? So it's really much more challenging. But on the other hand, if you have some idea about what you're looking for or what you're hoping to kind of detect, then you can make things better for yourself. But it's definitely much more challenging to do. This is why, yeah, to me, it's always good to do phantom experiments to test that your raw instrumentation is working. And you can also test certain other things that are experimental tests. It's still better, in my opinion, than simulations. But it's never the same as doing it in the clinic. That's much harder.
Okay. Do you have any input regarding the type of optimization used to tomographic image reconstruction? For example, sparse optimization versus typical L2 norm?
Yeah, so I mean, so again, we've tried L2 norm, L1 norm. We've tried, you know, for our breast imaging that we did, we worked, and I should say that really I worked very closely with Simon Arjun on this, who's one of the people, he's the founder of Toast, which is a sort of a commercial, it's not commercial, but it's publicly available software that you can get. Hamid Dagani also is an expert, and he has basically put together the near-fast software. It's also publicly available. They're closely related. But you know, we looked a lot. You know, so again, you do so for example, you can choose different norms for but you're basically still having some regularization parameter that you have to vary, and depending on the size of the regularization. So that, let me just, so if I describe that in physical terms, the regularization, this multiplicative parameter, it's determining how important how strong you want the regularization to be. And so there's a trade-off often between resolution and you know, sort of signal to noise. So there's something called an L curve, which helps you to choose. Again, this is a topic in its own right, so I'm not hopefully this at least will give you some things to look up. but you could look at this L curve and you can figure out the optimal regularization parameter for your system. And so, you know, normally, even when we were doing our breast reconstructions, we started out, we would do the same regularization for absorption and for scattering, but then we found that actually if we wanted to get sharper images in one feature, it was better to have different ones. So it's a game where there's a lot of parameters to play with, but I would only, you know, unless you're working in the field of image reconstruction, for me personally, I start to think about those problems when I have images that I want to sort of try to sort of pull out the most accurate, the most high fidelity tomogram from the data. But there are definitely a lot of different optimization parameters to play with, including the norm. But the big ones are the norm and the regularization, the strength of the regularization parameter.
Thanks for that. Yanis from Imperial College comes up with another question. Can a diffuse reflectance fiber probe follow the standard sterilization processes, such as autoclaving?
Oh yeah, yeah. The only thing for the diffuse reflectance, oh okay. So if you're doing NIRS or DOS, okay, so if you're just doing diffusive waves or this time domain analog of that, then yes. And also there's no, it can be multi-mode. It's not very, because all you're wanting to do in that case is figure out how much energy are you collecting in this area. So that's not a problem. But if you, for the diffuse correlation spectroscopy measurements, then you do need single speckles, or it's desirable. You can do it with multi-speckles, but it's better signal to noise if you have many single speckle detections. And in that case, you need a single mode fiber. And so then you can, again, you can certainly cleave it and stuff like that, but you just have to be more careful with that. Usually it's a lot smaller too.
Thanks. Adolfi from Ivory Coast asks, the light backscattered by a moving object and detected by a detector undergoes variations. How to determine the exact values of its optical cross-section, knowing that we have no idea of its size?
Maybe say that whole question again, sorry.
The light backscattered by a moving object and detected by a detector undergoes variations. How to determine the exact value of its optical cross-section, knowing that we have no idea of its size?
Yeah, so then that, okay, so if you want to figure out the total cross-section, so this is sort of a single scattering question. So, you know, again, just imagine it's a little bit simpler if we just have a dilute sample of the same kind of object. And you're shining, you can get the total scattering cross-section by just measuring the transmitted light through the sample and, you know, taking the ratio, the log of the ratio of the transmitted to the input light, right? And if you know the thickness of the sample, you can then determine the cross-section, and then you have to know the number of scatterers. So there is, you know, so if it was only one of them, you could in principle do that too, except that it will be a very small change, and so it might be really hard to measure. But if you have n of them and n is large, but not too large, so there's only single scattering, then you could do it that way I just described. If you measure the angular variation of the angle-dependent scattering, that's how you get information about its shape and size. The other way, you just get the total cross-section, and you don't really know much about the nature of the particle, except how much energy each one takes out of the beam due to scattering.
Mm-hmm. What are the advantages or disadvantages of multi-frequency versus multi-distance for absolute optical properties in the Fourier domain? Also, can you think of any other uses of varying frequency with FD and IRS?
Yeah. Okay, so that's a good question. And so just to remind everybody, so when we looked at the solutions of the diffusive waves, you remember that there was a dispersion, right? In other words, the wave vector, both the real and the imaginary part, depended not only on absorption, but it depended on the modulation frequency, right? And depended on D, the scattering. So so in principle, if you make measurements at a bunch of different frequencies, modulation frequencies, even for just one source detector pair, you can have enough information to, by comparing the transmission or the phase and the amplitude at the different frequencies, you have enough information to extract the optical properties. But that's only true if you use frequencies that are fairly high. Okay, you have to have that the frequency is, sort of in the at least comparable to the light velocity times the absorption factor. So if you can be in that regime, then having different frequencies is like having more measurements for the same source detector pair. And so ideally, it's better to have many source detector pairs and many frequencies. Some people, so you can do that in the frequency domain, like that's something that's done, that Bruce Stromberg has pushed for many years. And it's, but I mean, I would just say not all the frequencies have the same value. In a way, if you just have a low frequency and a high frequency, that might be almost as good as having many, many frequencies, right? As long as sort of one is below this V times mu A and one is above the V times mu A. So I would say the drawback of that approach is you have to have multiple frequencies. So the experiment is actually, the instrument is a little bit harder to make, but it's possible. The multi-distance, it has a lot of good things about it. But it does rely on the fact that the tissue is reasonably homogeneous underneath the probe. But nevertheless, I would say that if you all things being equal, it's better to have multi-distance and multi-frequency. I would say I would prefer to have if I had to make a choice between having multi-distance versus multi-frequency, I would take multi-distance. Because it sort of also gets rid of a lot of, it has a potential to get rid of some systematic errors too.
Okay, thank you for that. Next question is from Gary Rochester. In addition to tumor blood flow, can diffuse optic spectroscopy be used to measure reactive oxygen species, which might also be important clinically for monitoring cancer?
Yeah, that's a great question. So singlet oxygen would be the ultimate one, right? So Brian Wilson, who I guess is going to be talking or has been talking, he pioneered measurements of the lifetime of the, he pioneered measurements to figure out the singlet oxygen. I think that was a super challenging experiment. So in the current broadband NIRS or broadband DOS, it's hard for me to see that you're going to measure the reactive oxygen species. You can measure maybe if you're doing photodynamic therapy, for example, you can measure the dye. That's usually, because what you need is not only that there's molecules are living a long time, long enough to make a measurement, and that they have a distinct spectra, which could be the case. But again, you need a fair number of them, right? Because I mean, the fact that we can do hemoglobin spectroscopy, partly that's facilitated because there's so much hemoglobin. And so related to that, so again, this Ilias Tuxidas from UCL has been trying to measure cytochrome C oxidase, or the oxidized and reduced forms of that. And that's, you know, again, the spectrum, it's got some spectral features in the near infrared that you can use, but its concentration is pretty low. Okay, it's a lot lower than hemoglobin. And so small fluctuations in that situation, it's very, very challenging, although he's made a lot of progress and I think it's working. But basically, and the part of the reason it's challenging is because small fluctuations in stuff like hemoglobin or scattering could mask the changes you're trying to look for in the cytochrome oxidase. And I think you'd be in the same situation with looking for the reactive ion species. There wouldn't be that many. Again, that's if you want to do it in vivo, for sure. You could look at the effect of them on the dye, on the fluorophore or the contrast agent or the photodynamic therapy agent. So if you could figure out the effect of them on some other molecule that could be at higher concentration, then you could potentially detect it. So for example, another one of the things that I talked about in my fourth lecture was these oxygen-sensitive phosphors. Okay, and basically by looking at the lifetime of them, you can measure the concentration of oxygen nearby. Maybe there's a way that you could use those kinds of things to look at reactive ion species. But again, that's my speculation.
Thank you. Matasam from Sweden asks, how could the ratio of intensity of radiant energy scattered in a given direction to the incident irradiance be useful in the measurements?
I mean, so in the diffuse optical measurements, we're sort of using that idea, right? But it's like we're having, in our case, we're having a sequence of those events, and so you can't just, in that case, the angular information is lost. All right. So what you're describing, the question I think, as it was asked, is appropriate for single scattering, but in the multiple scattering limit, you can think of it as many single scattering measurements, but with all different kinds of input light angle scattering angles, right? And so you're averaging over all that, and so you know it's part of the problem, but you can't actually do that experiment directly in the tissue.
Okay, next question. In the extrapolated boundary approach, the choice of the height of the boundary related to A is important. Does this depend on anything other than index of refraction? Also, is there a good analytical way to find the boundary height?
I'm a little confused about what they mean by boundary height in this question. Do you know what they're saying? Can you say it again?
Okay, maybe I can read the question once more. In the extrapolated boundary approach, the choice of the height of the boundary in brackets is given like related to A is important. Does this depend on anything other than index of refraction? Also, is there a good analytical way to find the boundary height?
So the boundary height, I'm not actually sure what you mean by the boundary height, but I'm going to say you might mean the extrapolated distance, extrapolation distance. So that was LS in my slides. So that depended on the reflection coefficients, the Fresnel coefficients between the air, for example, between the air and the tissue. And in fact, I just wrote down some R effective, but basically it's very explicit. If you know the N of the air and if you know the N of both media, then you can compute, some kind of complicated integrals, but not that complicated, of the Fresnel factors to get what that LS is. And so the only controlling factor in that is the index of refraction. So that's my answer, if that was your question. So if not, feel free to write to me your email me, and I'll try to understand better the question.
Thanks, Arjun. The next question is from Farouk, ICFO. It's quite a long question. I'm likely asking a broad question, so please feel free to give more of an opinion-based answer. BCS and NIRS as suitable methods for the cerebral hemodynamics monitoring have been successfully integrated in the past with the EEG, which monitors the neuronal activity. Given some recent progress in EEG monitoring, namely ability to record extremely low frequency brain signals, what could that potentially mean for the possible improvements in the fields of DCS and NIRS?
I mean, okay, so there's actually, it's a really great experiment that you're describing where you're doing DCS, you're doing DOS, and you're doing EEG. So, and actually I'll say that, you know, the folks that are doing that, that's, they're leading the way and there are not that many of them. So I think almost anything that they find out, because what you can do with that measurement, of course, is you can connect really the electrical responses in your brain to the hemodynamic responses. So I think that's super valuable. And, but I think that what you were getting at about the low frequency may be that related to the networks. Okay, so again, I'll give a little background about this. So, you know, your brain has different areas that are for, you know, if I tap my finger, there's an area of my motor cortex that's involved in that. If you do visual stimulation, there's an area back here that's important for that. So generally, there's these different regions responsible for different activities in the cortex, but they themselves are have correlations and interactions with each other. And so these sort of networks of these sort of anatomical activation areas are called resting state networks. And what people have found, especially, well, people did this a lot of work with MRI, but people in the optical regime, it's really taking off now, both in animal models and even a little bit in human models. But if you look at the very low frequency spectrum, so way below the heart rate, okay, so there's a lot of power in there. It's not just noise. And the power in there has to do with fluctuations in hemodynamics, for example, if you're making hemodynamic measurements that are associated with these resting state networks. And so for one thing, I think it's really interesting to try to, so people are finding that they can identify these networks and characterize the coupling of different elements of the networks. And I think that you can do this as well, as I believe you could with EEG, is at least what I think you're asking. And that would be a very nice thing to try to compare. Okay, so I'll leave it at that.
Thank you. Next question. Can you explain on the ergodicity and on the validity of assumption of ergodicity in tissues?
Okay, so this is a question I think, again, more related to the dynamic light scattering, the correlation measurements, okay? So if you do a light scattering experiment in general, right? If you have, if everything is moving, so if everything is moving, then the correlation functions decay to 0. Okay, this is one way to say it. And if that's the case, then people call the sample ergodic, okay? And so like the time average of the measurement, in statistical mechanics, it has to do, ergodicity has to do with ensemble averaging being equal to time averaging, okay? So in practice, if all the scatterers are fluctuating, then your correlation functions decay to 0, and you're safe in that assumption. Okay, and actually what that does is it lets you analyze it more simply. But you could have a situation where part of the sample is static and part of the sample is dynamic. Okay, And that's certainly the case in general we would expect for tissues, right? But even for like gels, that could be true. And so then what would happen really is when you measure the decay of the correlation function, it may decay initially, but then it won't decay all the way to 0. It could be flat, or it could be flat for some time because everything's moving at some level, and then way later it would be it would decay to 0. So in that case, if you didn't know that the sample, if you didn't know that part of the sample is sitting still, then you could analyze. You have to be careful. You could misanalyze your data because you don't know where the 0 is, for example. And so anyway, so in the physics community, people figured out different tricks that one could do to get around this problem. And actually, so you could think, well, this could be a big problem for the tissues. But actually, I guess what the bottom line of it is that there is a lot of motion. Okay, and so the correlation functions we measure, they generally are going to 0. And so it's not, it's something that you would look out for, but it's not something that has turned out to be a big problem. But I'll say one more thing, and that is that the initial decay of the correlation function is the most valuable part of it, usually because that's associated with the deepest penetrating photons. And so people typically just use the initial sort of 1/2 or 1/e decay and don't worry about the decay all the way to 0. If you're fitting for the whole correlation function, this could be more of a problem. But if you're just sort of trying to pull off a time scale for the decay, then it hasn't been a problem for that people have had to worry about very much, and which is a lot of people at this stage, because evidently there's a lot of motion in the tissue in the end. So even the things that you think are static aren't really that static. So maybe the person who asked that question might appreciate some of the things I'm saying. It's kind of a technical point that maybe other people may not know about. It's a subtle point.
Thank you for that. Next question again has 3 parts, so I'll start with the first one. Does the finger structure satisfy the assumptions behind the diffusion equation? For example, source detector separation is around 1 centimeter, both in reflectance and transmission geometries, and the separations between boundaries and optodes are less than 1 centimeter.
Was it for the finger? The question was about the finger?
Yeah, finger structure.
So I think that the finger is on the border, all right? So I would, it's easy to use diffusion. I would use diffusion first. But then you may have to think about, you certainly want to do some experiments to think about how that might be corrected. And you might do some Monte Carlo. That might be the best way to do it. And so you may have to go, in other words, you may have to go back a little bit to the transport. But for the finger, I would try I would try to use diffusion. Because it's definitely probably mostly okay, would be my guess.
Okay, the second question is, what is the range of optical properties of optically thin media?
Well, okay, so when I say an optically thin medium, okay, so what that means, if you think about the beginning of my talk, we had a sample cell and light went through the cell. And I sort of said, well, what has to be true for this to be optically thin is that the light doesn't scatter. Okay. So it depends on 2 things. It depends on whether a medium is optically thin, depends on the L, and it depends on the scattering length, right? And so so I mean, it's hard to answer. So there are 2 factors that go into defining whether something's optically thin. So in other words, let me say it another way. So if I have something that's a centimeter thick, and I just have a medium so that I put polystyrene particles into it, And so then the light, and if it's a low concentration, maybe it's a very small amount of scattering, and the transmission might be decreased by 10% or 5%. But if I then multiplied the number of scatterers by 10, okay, then the transmitted light would drop by more than 1/E, right? And then I would say that's optically thick. But what you want to do is you want to figure out what's the random walk step in the sample, and what's the length of the sample, what's the length of the source detector separation. And if the length of the source detector separation is 5 random walk steps, then you're probably getting the diffusion approximate, then it's optically thick. It's starting to become optically thick. If the length is only one absorption, one random walk step, then it's optically thin. So that's the way to think of it.
Okay, the third question. In the time domain NIRS measurement, if a sample is measured by 2 different systems with completely different IRF width, how the zero time is better to be defined to provide more similar path length resolved information on the IRF maximum or on IRF rising edge?
Okay, so again, I'm not a super expert about the time domain. I would use the rising edge to define the zero for the 2 different systems. I mean, that's a question for somebody working on that problem, who's trying to set up that kind of experiment. Yeah.
Okay, the next question is from Yanis. How could a diffuse reflectance spectroscopy probe be translated to an imaging system in order to give higher spectral resolution, spatial resolution?
Sorry, could you repeat that? The whole question. Sorry.
Yeah, how could a diffuse reflectance spectroscopy probe be translated to an imaging system in order to give higher spatial resolution?
Yeah, so I'm going to recommend that you look at the work of Joe Culver, who's a professor in Washington University of St. Louis, and has been working on the brain problem to look at these resting state networks. And so the way to improve it, those measurements are in reflectance. And so what he does is he has first of all, you have a very dense distribution of sources and detectors. And then he does all sorts of tricks where he correlates, he looks at the correlations between detectors nearby, nearest neighbor ones and next nearest neighbor ones, and then ones that are far away, that help to subtract out noise. And so, I mean, it's certainly, images which are getting resolution, it's sort of quasi-topographic, it's tomographic, but it's, you can think of, maybe the resolution, like I said, that the depth of penetration was set by the source detector separation divided by, say, 2 to 3. And you might think that your resolution is set roughly by the source detector separation, then you're looking at just the region under that. So I think, by what Joe has done, he's done a really good job of getting the maximum resolution out of the diffuse optical system by having a dense pattern, careful pattern, and looking at correlations in the pattern very carefully in the reflectance geometry. So I think that answers the question. I'm not 100% sure.
Yeah, thanks. The next question is, slopes of phase and log RHO square AC, I think that's how it reads, are often used in FD and IRS to find absolute optical properties. However, both the zero boundary solution and extrapolated boundary solution show that these slopes are not truly linear, slope independent of distance. Are there other methods for retrieving optical properties with middle distance measurements without using linear slopes, maybe fitting to the complex reflectance from an extrapolated boundary?
Oh yeah, good, great question. So the slopes are linear only in the regime where you're many random walk steps away, where your detector is many random walk steps away from the source. So again, if the random walk step is a millimeter in the tissue and you're making measurements between 1 and 10 centimeters, then you're in a regime where it could be linear, should be linear. If you're not in that regime, which is often can be the case as well, then you just don't want to, you want to use this, first of all, you don't want to use that, there's an approximation to get it to be linear. So you don't have to use that. You can use the more exact solution and fit to the more exact solution. So there's a fair amount of papers early on that were written on reflectance, basically, that showed how to extract information in that regime, and definitely it's not linear. You're not fitting anymore to just a linear slope. You're fitting, then you're sort of going back to fitting to a function that is still predicted by theory, but it's not just a linear, and it becomes linear at longer distances, but it's not linear initially. So it's definitely possible to do that. That's a good question.
Okay, next question is from Hamburg student. In DOS, you mentioned we can find concentration of two substances, HP and HPO2 in your example, using two wavelengths. Can this in practice be extended to find concentration of N substances using N wavelengths? How many is realistic to be able to measure?
Yeah, the answer is yes. So in fact, normally if you're doing broadband spectroscopy with many wavelengths, you can also extract information about water, and you can extract information about lipid even, okay, concentrations in tissue. The key is the thing that really can help you, and the thing that helps you about hemoglobin is that it has a nice, pretty sharp spectra, right? So there are pretty distinct differences between oxy and deoxyhemoglobin. And actually water is pretty different too. So water is, adding water in is not a problem. I should say that I gave the example with two wavelengths and two unknowns. But in fact, also, it's much better if you had 5 wavelengths and 2 unknowns, right? Then your problem is overdetermined to some degree, and that's a good thing. And so you can usually get a better estimation, more robust estimation of the properties if you use more wavelengths, because you can spectrally constrain the problem through the extinction coefficients. So the answer is yes. But let me just make a comment. Like if it's N chromophores and N wavelengths, that's sort of the minimum. Actually, it's really N+1 you'd want because you have scattering. But it's better if it's like N chromophores and N plus N, 2N wavelengths or something, right? If you can do it that way. Thanks.
Thank you. The next question. Is there, compared to photoacoustics, a significant advantage using diffusion measurements to localize breast tumors? Would a hybrid system improve diagnostics?
I missed the beginning of the question, sorry.
Is there, compared to photoacoustics, a significant advantage using diffusion measurements to localize breast tumors? Would a hybrid system improve diagnostics?
I think that they could. The photoacoustic has the, in principle, it has the optical signature and the resolution, the optical functional information and the resolution of the ultrasound, which is improved. It has been, I would just say, for looking at, I mean, again, there's folks that were, Paul Beard, I think, was one of the lecturers, and he must have probably talked about this, and he's certainly much more of an expert than me. The quantification of oxygen saturation isn't all that easy. I mean, sort of, I would say that there's sensitivity, and I think it's even very interesting with looking at whatever sensitivity there is to see how good it can be to predict malignant versus benign and things like that with photoacoustic techniques. And I think it's very promising, actually. but it's not obviously done better at this point, I would say, because it's actually kind of hard to measure absolutely saturation and things like that.
Thank you. Next question. For in vivo experiments, can we integrate light modulation and demodulation in reflection probe using CW and IRS?
So what do you mean integrate? So once you put the modulation and demodulation in, I would just say that you're doing frequency domain, unless you're talking about really low frequency. If it's low frequency, then it's effectively still CW. So I'm not positive I understood the question, but from what I understand from what you said, that's what I would say.
Yeah, maybe we can ask him for some clarifications later and come back to it.
Anybody, by the way, let me just say that anybody who I easily may not have given a very satisfactory answer to many questions. So anybody who has any further question, feel free to send me an email.
Well, thanks. David McMahon likes to know what are your primary focuses in your own research nowadays? It seems you deal with a lot of areas.
Oh, okay. So well, first of all, we're full in about the brain, okay? But I'm trying to, like, I feel like in my life, I've gone from being, trying to just understand the basics of the technologies to, now I really want to get it to be valuable in the clinic. So for the brain research, we have experiments in the neuro ICU at Penn, at the hospital at the University of Pennsylvania. And we're basically trying to just do really longitudinal experiments for hours, right, at the bedside. And then you catch these events where there's, I gave a couple of examples of these, where there's some spike in the metabolism or some seizure or something like that. And we're trying to look at the signatures of that and to figure out, so like one goal would be, could we look at our properties that we measure and predict that a hypoxic event may be coming? Because that would be bad for the patient. So that's one thing, and you know, a similar, and that we have some good collaborations with folks in the Children's Hospital of Philadelphia, where they're, you know, doing these heart surgeries, and they're very injured babies and neonates, and we're wanting to look at their brain during these surgeries. How is their metabolism changing? To try to make predictions about whether they'll recover. To try to make predictions about whether they're, you know, some of them even have to go to ECMO, and so can we make measurements and then help to think about how to adjust the ECMO. Another set of problems that we're interested in about is measuring the mother's placenta, and that's a challenging problem because you have to have really deep penetration of the light. So for that one, it's not flow, it's just looking at oxygenation. And then with the cancer problems, I want to, you know, we're doing stuff that's kind of like machine learning. In a way, many statistical techniques that are used already, they just weren't called machine learning. But we're trying to use some of those techniques to apply them to do diagnostics for cancer tissues and cancer tissue response. So those are some of the areas I'm working on. Martin, you're muted. Oh, you're still muted. Still muted.
That was deliberate to be muted, but I did mean to say now that we have passed 5 o'clock. We do have 2 or 3 questions left, if you're happy to take them. And to remind people also, in 1 hour, we will start a Q&A with Brian Wilson from Toronto. So do you want us to finish out the last few questions?
Yeah, that's no problem. Be happy.
Okay. You mentioned ECBO, by the way. That's oxygenation outside the body for these little babies that are in need of that. Yeah. Okay, can't remember exactly where we stopped, but Wahajat from Oslo, in vivo experiments, can we integrate light modulation and demodulation in the reflection probe using continuous wave NIRS?
Right. So I think this was asked.
Yeah, this was answered.
That question was asked.
Sorry, so maybe Murali then, where do you see the future of DOS DCS? What sorts of improvements in source and detectors do you foresee in the near future that would move DOS and DCS to the next level?
Oh yeah, okay, so again, I see, okay, so I think one thing that is exciting in DOS is the cytochrome C oxidase measurements. So I think that's progressing really nicely from UCL, but it's still like we're actually trying to do it also, but we're collaborating with them. So it's I think that technology associated with that and the understanding of the systematics associated with that and demonstration of what works and what doesn't work, that'll have a good impact because it's another chromophore that's giving you molecular information about mitochondria. So I think that's an exciting area. In terms of technology, I think what could really help is to have stuff that's cheap and easy. Okay, and so you know, a big part of that is detection. Okay, and so there's been a fair amount of work using using ideas from the speckle contrast community to make parallel measurements cheaply with CCDs and fibers. And so I think that progress in that area could be good. I think what's going to have to happen there is you'll have to, for all of those experiments, if you really want to use those approaches, you're going to have to sort of realize something about your data, like how fast are they typically detecting, and then use that information to set something up. There's also some nice interferometric work. Vivek Srinivasan, I think, and he's been at UC Davis. I think he's moving to NYU, but he's done some really nice measurements in the frequency domain with interference, measuring the field, and he can do the time domain DOS as part of that, and he can do DCS as part of that, and even path-dependent DCS. So if you can gate, again, this is something I talked about in one of my fourth talk. If you can gate the photons to figure out what distance they traveled in the medium, that's the best way to eliminate the superficial tissues from the signals for the brain. And so I think that that's something that's maybe on the horizon. There are certainly a few groups working on it. It's challenging and expensive. So those are some examples.
Right, I guess there's a photon budget there that has to be balanced somehow.
Correct.
Last question then from Ireland. From Aaron, near the end of your lecture on clinical applications, you mention a few lower cost technologies that are being worked on. Would cost be the main factor that affects new technologies from being deployed? I guess that's what you're getting at in your last comment, into clinical settings, or are there other factors?
So I guess, and this relates to a question somebody asked earlier, that I feel like if we can prove, as researchers, if we can show that there's real clinical value in the measurements, then we can get people to make the fancy detectors, cheaper, and they'll spend more of their effort to do that because of the potential. I think to get stuff, our argument really is non-invasive, portable. A big part of our argument is non-invasive, portable. There's new contrasts for optics, and cheap is part of that to me. Cheap and easy is part of it. So I think all these, I love all kinds of technologies actually, but for getting it into the clinic, I think that's cheap and easy and accurate. Let's just say, and accurate enough.
And lastly, if I can just add to that or expand on that question, are the numbers there? Are there enough patients or enough doctors who would actually buy this technology to make sense if it did deliver?
So in my opinion, yes. I mean, first of all, just the regular commercial NIRS, right? Which is a CW instrument in almost every hospital that you go, right? There's a big market there, and so this would move into that market, at least and if you start to show it's good for looking at peripheral arterial disease, if you start to show it's useful for doing cancer, staging for neoadjuvant chemotherapy. I mean, I guess I feel optimistic about that.
Right.
I mean,
So
I'm not a businessman, but I have interest about it, certainly. But I think yes, and I would say that my colleagues, who are the consumers of this, they're super excited about it. When we're working on, like I said, for the brain, they're mostly drilling a hole and putting sticking some wires into the brain. If you can measure stuff like that non-invasively, that's a big bonus. They're super enthusiastic about just even sitting and looking at the data and figuring out what happened.
All right, that was excellent. Thank you so much, Arjun. We're slightly over time.
Yeah, so I'll just say once again, if I didn't answer your question, which is easily possible, properly or adequately, please send me an email to follow up.
Yeah. Okay. So that last point, I guess you're looking for the killer application that will pay for the development, and then the other applications can kind of come along under that umbrella.
That's
so
always
I suppose
the way things
yeah. So on that note, huge thank you to Professor Yodh from the University of Pennsylvania. My name is Martin Lee from NUI Galway, and this is the Biophotonics and Imaging Summer School, and we will be back in a little under 1 hour for Brian Wilson from Toronto. So thank you so much, Arjun. It's been a pleasure.
Thank you.
Appreciation is left there. We can't applaud exactly, but you will gather from the questions that they definitely appreciated your videos.
Yeah.
So
okay.
That's all from us.
Thank you, and so everybody have a good day.
Yeah. You too.
Okay. Yeah.
Bye now.